Yeah, sorry, that was a cat. So I think I'm at the wrong conference, I think. No, but yeah, we have actually a cat. And I would first say welcome to this this session about population dynamics, which I am quite fond of. And I mean, this is an example of a predator that is affecting a pest. So this is something I will talk about, but not about cats. I will talk about bark beetles and the population dynamics of bark beetles, and also their enemies. And I'm working on a very small scale. I'm working with, in this project, with single trees. So in Sweden, we have had a few outbreaks. And as you can see from the drought in 2018, it has increased extensively with 34 million cubic meter of dead trees. So I used this outbreak to study this. So in forestry, there is a conflict in conservation areas. We have the forestry, they are the flying bark beetles. From nature reserve, they are killing my forest. But then we have the other organization, like environmental organization. They say that no, but they have more enemies to control the bark beetles. So no one knows really what's happening, and it's also difference in forest structure and so on. From Wegensteiner, his excellent review, he say that they have about 170, or the IPS genus have about 170 enemies, and they can cause significant mortality of bark beetle populations. And the key taxa are woodpeckers and cleary beetles, the Panastimus, and some flies. And parasitoids are not considered to affect the bark beetle populations. So, and he also say that there is a complex interaction between different factors, and they vary in time and space. And yeah, the forest management has also an impact. So in Sweden, where I was working, we visited bark beetle killed trees during this outbreak. We were visiting it during 4 years. We were climbing up the trees about 3 or 4 meters. We took a bark sample of 45 times 15 centimeters. And when you have these kind of bark samples, you can estimate the attack density by counting the number of egg galleries. And if you also count the number of live bark beetles and the emergence holes, you can also estimate the reproductive success. And we took this bark beetle to the lab, and we tear them apart in very small pieces, and we pick out all insects that we can find in these bark samples. We were working in this area in southern Sweden during 4 autumns, and we took 222 bark samples, and they were widespread across managed forests, nature reserves, and set-asides, these woodland key habitats. And they are not really formally protected, but they are should not be used for tree cutting. And they have also conservation values, like large trees and deadwood and so on. So what we can collect from these bark samples, I mean, in the field, when we're in the field, we took some measurements about the tree diameter, crown color, and more. And when we took them to the lab, we can estimate the egg galleries and live bark beetles and emergence holes to use for the reproductive success, and also some natural enemy groups. We do not have the skills. I had a few master students, and they cannot distinguish species, so we put them in different groups, about 8 groups. And also we estimated the total biodiversity between these nature reserves, managed forest, and the woodland key habitats. Then we added some raster data from spruce, the area of spruce, and then we have in Sweden the soil moisture index, which estimate how wet the soil is. So we go directly to some results. So as you can see, we have the managed forest, and we have the woodland key habitats and the nature reserves. And on the y-axis, we have the number of egg galleries, which is the attack densities. So between the managed forest and these kind of three different forests, there was actually no significant difference in attack densities. So we looked at the reproductive success, and actually there was no difference between these groups in these different forests. So we continue, and then we looked at the natural enemies. So we had about 8 or 9 groups of enemies that we think can have some kind of effect on the bark beetles. And when we looked at a number of species, or actually species groups, we divided them in these 8 groups, we didn't find a significant difference. Do you think this is boreal? Or we can continue. And when we look at the enemy abundance, the total number of individuals, we found that it was significantly more individuals of enemies in nature reserves. So that was quite nice. And the output, that biodiversity, it was in woodland key habitats and nature reserves, they were marginally more in 2022. So we have some effect of the abundance of enemies, and it's probably caused by different kind of forest structures. One example is deadwood. We have the dimension of the fragmentation of deadwood and age and species composition of the trees. And I don't have my glasses, I can't read. Structure and density of stands. And then we have the landscape can affect the biodiversity. And more things that can affect this biodiversity and enemies. And it's also papers that state that nature reserves and the conservation areas, they differ, for instance, in the volume or of deadwood and deadwood types, and also tree species diversity. So it can have something to do with that. And then we look at our own data, what we took in these areas, where we took the bark samples, and we could see that the spruce diameter was larger in these conservation areas. And also the area of spruce around where we took the samples within 100 meters. They were higher in nature reserves. And then we have this soil moisture data that was significantly different from the managed forest. And I should say, when we look at the soil moisture index, it's when it's higher value, it's more wet. So it was drier in the nature reserves in comparison to the managed forest. And something was quite interesting, that was that when we took the soil moisture index and made a correlation on the attack densities, we have a strong, significant effect. And this means that here we have, when there are more beetles, when there is more water, the trees have a higher defense, and more beetles are needed to kill a tree. And when the trees are stressed and there is no water or very little water, fewer beetles are needed to kill the tree. And this is the attack densities. And attack densities are also very correlated with the reproductive success, because if you have a higher attack density, then the reproductive success, the number of daughters per mother, they are correlated negatively with the attack densities because of intraspecific competition. And this, I mean, you have also seen it in previous presentation from Anna and Mikko and so on. So the water is really important for the spruce bark beetle also on a very small scale. But here we have another paper from Müller, and they have looked at a very large area across Sweden on harvesting data. And I also found this in a previous paper. So we go back to the enemies and we look at the reproductive success. And we put all these enemy species groups as explanatory variables and see which species group can affect the reproductive success. So what we find was that the Plegaderus, the clown beetle, was negatively correlated with the reproductive success. And this has been seen with Mikko. Yeah, so Mikko, he claimed in his master thesis that this Plegaderus proved the most promising regulator and was highly correlated with the infestations. And yeah, it followed well the numbers of the IPS to Graphus. It's also a study from Canada, I think. And they have looked at IPS Calligraphus, and they found that another species of Plegaderus, they can reduce or increase the mortality of this IPS species with about 15%. So maybe this is a new predator in the class of Meditera and Thanasmus, or it could be correlated with something else. Maybe this is just a causal effect. They like the same environment or habitat. So shortly, the dynamics of the spruce bark beetle, they were not really influenced by the forest conservation. And this densities and the reproductive success, for instance. And forest structures and condition, they differed between conservation areas and managed forests. And we also have more, the abundance of natural enemies was higher in nature reserves. And this may affect the reproductive success. So we have this correlation with the Plegaderus. So, and the soil moisture was also playing in this study, as it affected the attack densities, which is correlated with the reproductive success. So in the future, I would like to add your remote sensing data to understand also the beetle pressure in this study. So this is a wrap up of my conclusions. We have some factors that is affecting both the population dynamics and the enemies. I think, yeah, and in accordance to what Wegensteiner say, that there is a complex interaction between these factors, and the forest management is also a player in this game. Thank you.